Hello, my name is Diego Cardeñosa, and I'm an assistant professor at Florida International University. And today I'm going to walk you through the process of using the DNA protocol, which is a very quick and very easy step-by-step protocol to identify any shark species in any products that you can come across, either dried fins, processed fins, skin, meat, either processed or dried or frozen meat. It should be able to identify any shark product that you come across. So let's get ready, let's put our gloves on, and let's walk through the step-by-step of how to use this tool. First thing we're gonna do is to wear gloves to make sure everything is clean. All right. And we have to clean the surface that we're gonna be working on really, really well. So here we have alcohol. I'm just gonna be pouring alcohol on top of the surface. And then just wipe it out with paper towels. Make sure you clean every inch or every centimeter of the table. This is to avoid contamination when you run your samples. So it's very important that you work on a clean surface. Look. So it's very important to have it. Maybe just a little bit more. All right. Now we're ready to make the connections, take out all the equipment out of the bag. So here's our Pelican case. Just gonna open it. And we're gonna remove this foam carefully. Remove the machine out of the Pelican case. You can also take out the connectors, the chargers, the electronic pipette, and the small pipette. All right. Get this out of the way. And let's make all the connections necessary to turn on the machine. So this goes here. I'm gonna plug this here in the back. Done. Now, make sure that the machine is turned off, and then you are gonna plug this here in the back. Make sure the Bluetooth or Wi-Fi antenna is on the mic, and then you can turn it on. I'm just gonna turn this. As soon as you hit on, a blue light is gonna appear in the front of the machine. That's it. That means it's on idle and it's ready to be used. All right, now we're gonna get our reagents out and our samples out to get the DNA extraction part of the protocol. So here, I'm gonna put our samples there. All right. So today we're just gonna do a couple of fins for this training. So I'm gonna be doing 2 fins for this training. So we take 2 tubes out. Here on the reagents we have something called quick extract. I have it here as an aliquot. And it's the buffer that we're gonna use for the DNA extraction. So now you're gonna take a paper towel, place it here, take one of the samples that you're gonna be analyzing. So for this one, we're gonna use this. Place it here. Take a blade to cut the fin. Throw it away. And I usually take the sample from, you can see this fin is all cut off here. I usually take it from this edge of the fin, or either from the base or this edge of the fin. It makes a cleaner cut when you're dealing with either small or big fins. So I'm gonna make the cut, and it's gonna be super, super small. So think about a third or a quarter of a grain of rice. It has to be something really small. If you add too much, you're gonna be overwhelming the reaction. So it has to be really, really small amount of the tissue. Oh, I forgot one thing. So before you clean your fin, one thing you need to do is to clean the fin, because sometimes when you're analyzing this type of samples, they've been in contact with other fins in the same bag or in the same container. So you need to clean them before you cut them, just to remove DNA from different species that might have been in the container. So pour a little bit of alcohol on a tissue paper and just clean the area of the fin that you're gonna be doing the cut. Just clean it with alcohol, and that should be enough um, to cut your samples and not have extra contamination. So I'm just gonna make the cut here. And as you can see, it's a really, really small piece of fin. You don't need anything more than this. You're gonna open one of your tubes and drop it in, close it, and then you can discard this blade because you don't want to have cross-contamination from this sample to the next one. So just discard that blade and put your sample away. This paper towel also goes away, and then you use a new one for the next fin or the next sample that you're going to be cutting. Then we have this one. Again, you place it here. You take a piece of paper towel, drop some alcohol in it, and then clean the location in the fin that you're going to make the cut. Same as with the other fin. And same with all the fins that you will be doing your inspection, whether there's 1, 2, or 100. Every time you have to clean your fin or your sample really well before you make the cut. Take another blade and make the cut on the fin. You can see the size is very similar to the one that we just previously did. So small, half or a third or a fourth even of a grain of rice. It's really, really small. Then open your tube, place the sample there and close it. Discard the blade, take your fin out of the way, and then just do this as many times as you need, depending on the number of samples that you're going to be analyzing. Now we're going to take this pipette and we're going to do 20 microliters of the DNA buffer. So use the purple and small pipette tips for this process. Open them up, 20 microliters. Open your buffer. Take the 20 microliters and you're going to place it in your tube. Shake it a little bit and make sure that the sample is embedded, is inside the buffer. So make sure the sample is in contact with the buffer inside the tube. And you're gonna count at least 10 minutes from now. Discard the tip, and we're gonna make here a timer for 10 minutes at a minimum, where that sample has to be incubated in the buffer. So let's do the next one. I'm gonna change the tip here. 20 microliters of the buffer go in here in the second tube. Shake a little bit and make sure that the sample is in there with the buffer. And I'm gonna take a third tube and I'm gonna add 20 microliters of the buffer, just the buffer, and that's gonna act as my negative control. So one thing is very key and you need to do is always keep a negative control through the entire process to make sure that your reagents are not contaminated and compromised. So every time those negative controls have to be negative in the test, and we'll see that once the whole process is finished. So take 20 microliters of the buffer into a new tube, and that's just not gonna have any sample, just gonna be the buffer by itself. But it's gonna let us know that the buffer that we're starting with to do everything, it doesn't have any DNA, it's not contaminated with anything, and that we can trust our results once we have them. And now we're just gonna have to wait for 10 minutes until the next step. So for this test and this trial, we're just gonna do 2 samples. And you just have to replicate everything we do with these 2 with as many samples as you're gonna be doing. So the first thing is you put your Eppendorf tubes on the rack, and you're gonna label them however you want to label your samples, from 1 to 48 or from 1 to, or with a code, whatever you prefer. I'm just gonna label this 1, I'm gonna label this 2, and I have a third tube here that is gonna go for the negative control. So I'm gonna label NC as negative control. And now we're just gonna add the buffers, add the samples to each of the 2 tubes, and then wait for 10 minutes until the sample is incubated in the buffer for the DNA extraction. 10 minutes have passed by. So the 2 samples here should have been incubated and we should have DNA in there already. So grab your MIC tubes, which are these little tubes here. Just open them up. And we're gonna add 15 microliters of that buffer that is in those Eppendorf tubes into the MIC tubes. So grab your pipette tip Open your tips, grab one, take sample number 1 that we just labeled, open it up, take the tip to the bottom, take 15 microliters out of it, and then you're gonna place it into the first tube of this rack. How do I know that's the first tube? If you see here, all the tubes, they come in groups of 4. In each group of 4 has one tube that has a little notch at the top on the left-hand side of the tube. So that means that is the first tube of the group of 4. So use that as a reference to keep track of where you are. Now, sample number 2, pipette, take a new tip, and you're just gonna take again the 15 microliters out of this one. And then put it in the second tube, which is one below the one that you just did. Right? Discard the tip. Now for the third tube, you're gonna add the negative control, which is just the buffer without any sample. And this is going to confirm that that buffer is not contaminated by the time you're analyzing your samples. So open it up, take 15 microliters out of it into the third tube. And one thing about this machine is that it works as a centrifuge. So you're always gonna have to, even if you're running 1 or 48 or 20 or 24 samples, all the rest of the wells have to be filled with tubes with the same amount of volume that you're running your test. So for this one, we have 3 tubes with 15 microliters, but there's one empty. So that one has to be filled up with water. And we're gonna fill up 15 microliters of water just to have the same amount of volume in it. So here's water, just normal water, purified water. And I'm gonna add it to the fourth tube. Done. Discard the tip, put this somewhere else, and we're gonna close those tubes with the caps. These are these white caps that come with the machine, and this little tool that is a clamp to close the tubes really well. So we're just gonna grab one. Try not to get your hands in there so you don't contaminate all those caps with your hands. So just try to bring them to the front, take the clamp, grab four caps like that. They also come in groups of 4. And then you're gonna line them up with your tubes like that. And then once they're lined up, then you're just gonna press hard to close it well. And then you just release the clamp, and then you're gonna have your tubes closed and ready to go. So now we're gonna be using the mic. Okay, so here is the machine and the wells. So you take this magnetic clamp out of there. You can attach it to the lid, and then here it tells you where well number 1 is in this circle, which is right here. This is well number 5, number 9, number 13, and it actually shows you also where the little notch of the first tube is. So remember, your first tube, the first sample that we added, had that little notch on this side. So that goes on the inside of the circle and in well number 1. That way the software identifies this one as well number 1, this one as well number 2, this one as well number 3, and this one as well number 4. Because we're running only these 4 samples now, 2 samples, the negative control and the positive control, and everything else is empty. We cannot run the machine empty. We always have to add tubes that are filled up with the same volume of the reaction that we're running. So all these tubes are filled up with 20 microliters of water. So it evens out the volume of the reaction, and we have to fill up the whole thing every time we run the machine. If you're running 8, then you add your 8 tubes here, and then you replace them with the rest of the blanks. Then once you have them all ready and pushed down, put back the clamp on top of them, close the lid, and then you're ready to go to the computer and get the machine running. Now take your laptop out of your bag. Going to the software that connects the mic to the computer. Here it says that no instrument is found. Click on the icon of the mic with like a Wi-Fi signal next to it. Click on this icon right next. It says no instrument found, but the machine is on because we're seeing the blue light here. So you just click on that icon and it's gonna try to get the signal of the machine. Now it has it. It has the name of the machine. This one is called James Bond. And it says that it's on idle. So now the computer and the machine are working together, are connected. The next thing is you're gonna go on new, click on new, click on run, new run. Here where it says assays, you're gonna click on the plus sign, and there's one there that should be called quick extract. Then here in samples, you can label your sample 1 as 1, 2, and then the third one is your negative control. Negative control. Now you're gonna click where it says the name of the machine, and then you're gonna click on start run. You have to label your the file name. I usually go for quick extract, which is just the extraction of the DNA. You save it, and then you hit start, and you're gonna see that the light of the machine passes from blue to green. When it's green, it's because the machine is running, and then when it's a blinking green, that means the run was successful and it's over, if the green is blinking. So if it's red, that means something went wrong and the protocol or the procedure was not carried out to completion. So start, the lid is locked, you hear that sound, and then the light has turned green. This procedure says here is gonna take 9 minutes. So while those 9 minutes run, we're gonna start preparing the PCR to put those DNAs into the PCR mixes. The first thing we're gonna do now is change gloves. All right, so I have new gloves now, and this is also a process that has to be very clean, very tidy in order to get it successful. So I'm gonna take again a clean Eppendorf tube, put it in the rack, and we're gonna do the mixture for the PCR. The PCR has basically 3 components. One is called the Melt Doctor Master Mix. That has everything, all the reagents, everything combined into one thing. Every reaction or every sample has to have 10 microliters of the Melt Doctor. The other component is water, molecular grade water that you can get in these little tubes, basically anywhere. Wherever you run or you order your primers, they should be able to sell you little tubes with molecular grade water. And then the other component are the primers. So here we have a forward and a reverse primer in tubes that I have previously labeled forward and reverse. They're still a little frozen. And that's all the reaction needs. Melt doctor, forward primer, reverse primer, and water. So the reaction has, one single reaction has 10 microliters of the melt doctor, 3 microliters of water, 2.5 microliters of the forward, and 2.5 microliters of the reverse primer. That gets you to 18 microliters, and then you're gonna add 2 microliters of the DNA extraction into your reaction, and that's a 20 microliter reaction. That it's all you need to run these tests. So because we're gonna run 4 reactions, one for sample number 1, one for sample number 2, one for the no negative control, the no template control, and another one, the fourth one, for the positive control, which in this case is something we call a G block or the DNA positive control. So this has a synthetic copy of the DNA of a silky shark, and that's gonna give us the positive control. And you're gonna add 2 microliters of these to that reaction. So you're gonna have to multiply everything by 4. But because sometimes the you might do a little bit of a mistake, or the pipettes are a little off. So you never calculate exactly for how many tubes you need to run. You calculate a little higher. So for this example, we're gonna do 6 or 7 samples. So I'm gonna grab the electronic pipette. This one has already some programs there to run PCRs for 48, for 8 samples, 32 samples, 24 samples, 16 samples. So I'm just gonna use the program that has 8. So the first thing is gonna tell us to add one of these tips, and it's gonna take a volume of the melt doctor. So the melt doctor is already melted. It's gonna give it a little shake here like that. Open the tube, put the tip inside the tube, press the trigger of the electronic pipette, and that's gonna take the volume that you need. Then you're gonna click back inside your clean appender tube, and it's gonna dispense the volume with usually an extra volume in the pipette tip, which you're going to discard inside your melt doctor tube. Close it. Now you have to change this tip. The program in the electronic pipette should have a step to discard the tip. So you press the trigger and the tip should pop off. Next thing you do is get a new tip, and then this is for water. So again, take a new tip, place the tip inside the tube, press the trigger once, takes volume, press the trigger once inside this tube, dispenses, there's an extra volume there, discard. Now, because this water should be clean, you don't need to change this tip. You're gonna go into your forward primer. This one's still a little frozen. Just hold on one second. All right. So open the primer, insert the tube, the tip inside the tube, inside the liquid, press the trigger once, it takes a volume, then you take that into your Eppendorf tube here, press once, and then discard. Now you have to change the tip. Here is program to change it. Press the trigger once, it spits out the tip. Take a new tip for the reverse primer, which is also frozen. Done. Open it up, insert the tip inside the liquid, press the trigger once, sucks volume, then come here and press the trigger once, dispense it, and then discard here. And now make sure everything is at the bottom. And the pipette tip has the pipette has a step where it takes volume and dispenses it quickly, so it mixes everything up inside the tube. So take the tip of the tube of the pipette tip all the way down to the liquid and press once, and it's just gonna take in and out liquid to mix everything in the tube. So that's it. Purge. So the next step is go back in to the bottom, press once the trigger. It's gonna suck up all that volume. Just hold on 1 second. And we're gonna dispense 18 microliters into 4 tubes, clean tubes that you have here. So 1, 2, 3, and 4. If there's any extra, you can just put it back in the Eppendorf tube, discard the tip, and that's all you need to do. Close this just to keep everything as clean as possible. Put your reagents back here so they remain cold. And as you can see, the machine is already blinking green. That means the reaction is done and everything was successful. In the computer, you can hit here OK in the announcement that says the run completed successfully. So you click OK. And you open up this. Once you open up this, the light is going to turn blue again. You remove the clamp, put it in here, and then you take this, go back to your samples, which are these ones, and then you have to take off all the dummies or all the blanks that you put in there as well, to make room for the other ones. All right, put the clamp back on, close it. Now here we have your 4 tubes loaded with the 18 microliters of the master mix. Everything's ready. Now you need to add 2 microliters of the DNA into each one, and then we're gonna be ready to finish the test and let it run for an hour and a half. So I'm gonna set this pipette to 2 microliters. And then take your samples. Again, sample number 1 is the one that has the notch on the tube. You open them up carefully. And then take a tip. Go all the way down to the bottom of your first tube and take 2 microliters. And then this one goes to tube number 1 where you have your master mix. So go in, dispense your DNA, take the pipette tip out, and discard. Next one, tube number 2. Take 2 microliters, go to the second tube, dispense your DNA, and discard. Then on the third one, you're gonna have your negative control. So take 2 microliters of that into the third tube. this card. Then you can save this DNA if you want to follow up with sequencing, or if you want to leave it for evidence or anything. You can save them and keep them in a fridge at 4 degrees Celsius for weeks to a month. If you want to leave them store them for longer, you can just put them on below minus 20 degrees or freeze them, and then that should be stable for a long time. And in the fourth tube that we still haven't added anything yet, we're gonna add what we said before, that is the positive control. So we take it out, open it up, take 2 microliters of that, dispense in the fourth tube, discard the tip, close this, put it back in. Let's go ahead. There you go. Now we're gonna close those tubes with your caps and your little clamp. Again, don't go in with your fingers, just keep the clamps. Just use the clamp to grab the caps. Align it with your tubes, press hard down. Hard, you don't, I'm just making a lot of force right there. Remove the clamp, and then your tubes are good to go. Make sure you close these ones as well. Give them just a little shake, and then open your machine, remove the clamp, go to number 1 here again. You see the well has 1, 5, 9, 13, 17, 21, and that's the number of each of the tubes that have the little notch. And actually the notch is also portrayed in the icons in the well. So this one is gonna go in well number 1. That means this one is in well number 2, well number 3, well number 4, and the next one will be well number 5, which is labeled right there. And I have dummies or blanks for these ones as well. So these ones have 20 microliters of water. So I'm just gonna fill all the wells with it, because you always have to run the machine full, regardless if you're running 1 sample, 20 samples, 30 samples, or the full thing. Done. I'm gonna close again the clamp, close the lid, and you're pretty much done. So the next thing, you go back to your computer, hit new, run, click on run. On the plus sign on assays, you're gonna go to DNA elasmo. DNA elasmo. Hold on. Let's do that again. I think I clicked twice. So new, run, assays, DNA elasmo. Gonna go to samples, label your samples 1. The next one is 2. The next one is your negative control. And the fourth one is your positive control. Then you go to the name of your machine, click, start run. You're gonna label your run with whatever name you want. So we're gonna put the name training, save, and then you're gonna click start. Again, it turns green. It makes that loud noise. It spins the samples. It says here centrifuging samples, so it spins them. So everything is at the bottom with no bubbles. Then it's going to incubate the samples. If you hit here in run profile, it tells you what it's going to do. So it's gonna incubate the samples at 95 degrees for 10 minutes, which is this portion. Then 95 degrees for 15 seconds. It lowers down the temperature to 56 degrees Celsius for 30 seconds. Then it raises the temperature a little bit to 72 degrees Celsius for 30 seconds. And that's one cycle. Then it goes all the way up to 95 again for 15 seconds and does that up and down of temperatures 35 times, 35 cycles, which is everything you see here. And then it's going to incubate, after the 35 cycles, it's going to incubate the samples at 72 degrees for 5 minutes. Then 95 degrees Celsius for 15 seconds, 60 degrees Celsius for a minute. And then from 60 degrees Celsius to 95 degrees, it's gonna start incrementing the temperature from 60 to 95 at a rate of 0.1 degrees Celsius per second. And that's gonna give us that melting curve that we want to identify the species. The computer here in the software tells you here the time remaining, which right now is 1 hour and 36 minutes and 25 seconds. And that's the time that we're gonna have to wait to get our results. And then once you're done with this, discard the tubes that you already used, put everything back where in your bags or in your wherever you're carrying everything. So you have your station cleaned and ready for, once the machine is done, you can read the results or you can pack everything and leave. So use this time to clean everything, pack everything, and make sure everything is where it should be before you leave for your office or where you leave for wherever you need to go. So now the test is done. You can see the machine is blinking green. That means it was a successful test, and everything was conducted according to the parameters of the assay. So now I just clicked here, okay. That means that I acknowledge that everything was ready. So if you go here to in the analysis portion of the software, on the plus sign, and you hit DNA Elasmo, it's gonna give you 3 different curves. So I'm just gonna click here and select none. That means there's nothing on it. So for the positive control, this is what you should expect. It's always gonna be the same curve. It's always gonna have this shape here, this kind of like plateau that comes up and then comes straight down to the bottom. In the cycling, you can see this is the positive control. And then if you click it out, you can see that the two samples, sample number 1 and sample number 2, were shooting up. That means the reaction occurred in each of the samples. And then here you can see the no control, and you can see that there's no curve shooting upwards. That means there were no reactions in the negative control, which means all our reagents are clean and that we can trust our results because yeah, the reagents and everything were not compromised by any outside DNA, and they were not contaminated. So when you hit here on the DNA elasmo under melt, this is the positive control, and then you can see, you can click on the sample number 1, and here you can see that there's only one peak, and there's an inflection point on the ascending side right here of the curve. So if you follow the dichotomous key for identifying the curves, it's very straightforward. So in step number 1 of that key, is gonna ask you to go to the melt curve and see the number of peaks. It can be either 1, 2, 3, or 4 peaks. This one has 1 peak, clearly is 1 peak. Then it tells you to go to step number 2. Step number 2 says go to melt curve again, so you stay there. And is the tip of the peak, the high point of the peak, is at or before 77 degrees Celsius, or is after 77 degrees Celsius? So if you put your arrow of the mouse on top of the peak, you see down here that it says 78.46. So that's after 77. So you go to point number 7. That also allows you to stay in the melt curve, and it asks you whether there's an inflection point or plateau on the descending side of the peak, of there no inflection points on the descending side. You can see here the descending side of this peak is pretty straightforward. There's no inflection points. It just dropped from the top to the bottom as a straight line. So there's no inflection points. Go to the 13th point. That one tells you to do high resolution melt. So click here on the plus sign, then here on DNA elasmo. For this one, when you do the high resolution melt curves, in the reference genotypes, you have to assign as a reference genotype the positive control. Here you can name it PC, positive control. Then you hit this curve here, the second icon to the right, then difference to PC, and then you can see 3 different curves. One is for the negative control, which you can unclick, and there's 2 other curves right here. We're analyzing the curve for the first peak, which is this one, the first sample. And then for high resolution melt, it says if there's a valley before the tallest peak, or if there's no valley before the tallest peak. So here in point 13, it says if there's a valley before the tallest peak, or if there's no valley before the tallest peak. You can see here there's a little drop in the curve, a little valley, a little, valley before the tallest peak. So you go to point 14, it asks you if the valley depth is approximately half or greater than half the height of the tallest peak, or if it's like a shallow dive or a shallow valley. This one is quite a shallow valley, is significantly less than the height of the tallest peak. So you go to point 18, which it asks you to go back to the melt curve and asks you if there's a plateau on the ascending side. You don't see any plateaus here. So no plateau on the ascending side of the peak. Go to point 20, which stays on the melt curve. Asks you if there's an inflection point on the ascending side of the peak. Here you can see the little inflection point. Go to point number 21, also on the melt curve. It asks you if there's an inflection point located at the base of the peak, or if there's an inflection point located halfway down the ascending side of the peak, and that's the one. It's right here. So that gets you that this is Carcharinus falciformis. This is the curve that you would expect if you see a silky shark, or if you are analyzing a silky shark sample. But then if you see the one for the second sample, it's quite different. This one has 2 peaks right at the top of the curve. So let's go back to point number 1 in the key, just to check what's there. So this time 2 peaks. It asks you to go to point number 24. Then it asks you if both peaks are the same size, if the first peak is smaller than the second peak, or if the first peak is taller than the second peak. In this case, the first peak is taller than the second peak. So it asks you to go to point number 48. Point number 48, it asks you to go to melt curve, to high resolution melt curve, which is this one. Whether you have 1 peak or 2 or more peaks, this is clearly just 1 peak right here. Go to point 49. In high resolution melt curve again, is there a valley below baseline after the peak, or if there's a valley just under or at the baseline, or no valley at all, which is this is the case for these species. Then you go to point number 58, go to melt curve. If the first peak is after 77 or before 77, this one is clearly after. So go to peak 259. Again, melt curve, second peak short, reaching below half the height of the first peak, or second peak is tall, reaching above half the height of the first peak. I would say for this one is below half the height of the peak. Then on point 60, it asks you in the Mel curve if the first peak is or after 78 degrees Celsius, or if it's before 78. This one is right before 78. And then in point 63, it asks you if there's an inflection point close to the tip of the ascending side of the first peak, or if inflection point is not close to the tip of the ascending side of the peak, or if there is no inflection point. In this case, no inflection point, go to point 64. Again, slight or no inflection point on the ascending side of the first peak, go to 65. So for the second peak here in the Mel curve, it's clearly two peaks at the top. So from the first point, you jump to point number 24. There it asks you if both peaks are the same size, if the first peak is smaller than the second peak, or if the first peak is taller than the second peak. Here you can see the first peak is taller than the second peak. So you move to point number 48. In point number 48 on the high resolution Mel curve, when you move there, you can see there's clearly just one single peak. So it directs you to point number 49. In point number 49, it asks you if there's a valley below baseline after the peak, or if there's a valley just under or at the baseline, or no valley at all, which is the case. There's no valley after the peak here, which you go to point number 58. In point number 58, it asks you to go back to Mel curve. First peak after 77 degrees, or first peak at or before 77. If you put your arrow here of the mouse, you can see it's 77.74, so it's after 77. So go to point 59, which asks you on the Mel curve if the second peak is short, reaching way below half of the height of the first peak, or if the second peak is tall, reaching above half the height of the first peak. The height of the first peak is considered this whole thing. So this one is clearly above half the height of the first peak. So it makes you go to point number 66, and then go to Mel curve. The first part says two peaks about the same size with a shallow valley between them. So there's these two almost the same size here at the top, a very little valley in between them, and it tells you that that's Carcharodonus acronotus, or the black-nosed shark, which is correct. This is the curve that you should expect if you have a black-nosed shark in one of your samples. So this key that you have is just a temporary solution to identify many of the curves, or all the curves that are designed in the protocol as of today. But we're working on a machine learning technique that will be automated. You only need to submit the output file of the MIC, this file that you are analyzing here, submit it to a web-based or a software, and will automatically give you, with a 99.2 accuracy, each of the wells, each of the samples, which species does it correspond to. So that's in the works. It should be ready fairly soon. In the meantime, we have to use these curves, or get used to the shape of the different curves of the most common species. If I see a silky shark or a black nose curve, I know exactly what it is without going through the key. I know exactly what species it is because I'm familiar with it. But until you get familiarized with it, I recommend you that you go through the key, and it should always lead you to the right answer for each of the curves. All right, so that's it. As you see, the process of extracting the DNA, cutting the sample, putting in the buffer, putting back in the MIC, and then mixing a couple of reagents is very simple. You just need to get used to the pipettes. That's it. And then the machine does it all. Does the rest of the procedure. Then you get this type of curves. They're clear signature of each of the species that we have been able to analyze so far, which is 66 different species. That number is going to increase. And as I said before, we're working on a model that has so far a 99.2 accuracy reading these curves. So you don't need to go through the key in the future. You just need to upload the output file of the software of the MIC into a cloud-based platform, and it will give you the answer for every single sample in the protocol. That will speed up the process of analyzing it and not going through each single curve. But in the meantime, you can get used to the curves of the most common species. So for example, these two, if I see a black nose or a silky shark curve, I know immediately what it is because I'm familiar with it. So just use the key until we'll give you the software or the cloud-based platform to analyze them, and then you'll be ready to analyze up to 46 samples at each time. Remember, every time you run it, you have to have a well reserved for the positive control and a well reserved for the negative control. That allows you to make sure that you can trust your results. So always reserve two of the wells, one for the positive and one for the negative. And besides that, that's it. Just try to follow the steps of this video. Try to follow the step-by-step guide that we also provide you. And you should be able to go through the whole process by yourself. Both Daniel and I are available. If you have any questions, you can email us, and we can answer any of the questions that you have. And we hope this really helps you to identify any shark products that come across your inspections. Thank you.